Hi, I'm Jeffrey Townsend at Yale University in the Department of Ecology and Evolutionary Biology, and I'm here to tell you about a paper we've recently published in PLOS One, Evolution of Reproductive Morphology in Leaf Endophytes. Endophytes are organisms that live within host plant tissues, usually without causing any disease or symptoms. Fungal endophytes can be found in diverse parts of a plant, from underground root tips to leaves on branches high in the air, from the inner heart of a tree to the outer bark, from young shoot tips to the tips of old widowmakers, from primitive mosses to flowering plants. Diverse fungi occupy endophytic roles in a diversity of plants. For instance, species in the fungal genus Glomus can grow within grasses, forming endomycorrhiza of grasses. Fungi within the Basidiomycetes, which are mostly mushroom-forming fungi, inhabit tree roots, emerging within the soil as ectomycorrhiza of trees, such as this chestnut tree. Fungi from both the Basidiomycetes and Ascomycetes form orchid mycorrhiza, such as pictured here in root cross-sections of Cephalanthera longifolia. Fungi from the Ascomycetes and Leiotiomycetes form ericoid mycorrhiza, pictured here in roots infected with fungal endophytes colonizing a species of rhododendron. A curious example that you may have seen on a hike or a walk in the woods is the ghostly and colorless plant referred to as the Indian pipe, which can perform no photosynthesis, but is instead a parasite on other green plants via underground endophytic mycorrhiza.
Fungal saprobes, in contrast, make their living consuming dead tissue, such as this fallen log. Generally, the reproductive morphology of both endophytes and saprobes is the most distinct observable morphology for fungal species, because most fungal species spend most of their time growing asexually as nondescript hyphal tubes. For this reason, reproductive morphology is highly important to fungal species description and to determining relationships with other fungi, and is the most interesting morphology in fungi for studies of organismal development. Nonetheless, above-ground fungal endophytes of plants were understudied because their collection depended upon chance observation of their diminutive reproductive bodies at transient stages of their life cycle, or upon their ability to be cultured on artificial media. Polymerase chain reaction techniques now facilitate their rapid identification, and in fact,
examination of DNA markers demonstrates that a single tree might host several hundred or more fungal species of endophyte. Here, this diversity is demonstrated by the different banding patterns observable in each lane of the gel pictured. Almost nothing is known about how the above-ground fungal-plant relationship and its diversity have evolved. In our paper, we show that in the fungal clade called the Leiotiomycetes, there is extensive variation in reproductive morphology and size. Additionally, species vary in ecological role, from leaf endophytes to ground-dwelling saprobes. By mapping reproductive morphology to ecological role onto the evolutionary tree of the Leiotiomycetes, we show that leaf endophytes tend to produce small, highly reduced fruiting bodies, while saprobic species produce large and intricate fruiting bodies. To demonstrate these results to you, we'll first show you some anecdotal examples of endophytes and saprobes within the Leiotiomycetes that are consistent with the trend we've observed. Then we'll show you a more rigorous phylogenetic analysis that accounts for the correlations caused by common ancestry and demonstrates that the trend represents a strong correlation that is statistically significant. First, let's describe what we observe about the trend of leaf endophyte sexual reproductive morphology, showing the reproductive morphology for several exemplars. The sexual reproductive body is usually small, sometimes so small that it cannot be seen by the naked eye, as in this species, Taphrina weissneri, causing leaf curve on peach trees. Most leaf endophyte sexual reproductive bodies are also covered by a hyphal tissue from the fungus, or by dead sclerotinized tissue from the host, as may be seen particularly clearly in Febrella tsugae, a conditional pathogen on conifers. Lastly, usually the outermost layer of the sexual reproductive body is covered with dark-colored, thick-walled cells, as may be seen particularly clearly in Rhytisma americana, known as tar spot, mostly inhabiting maple trees in North America. Next, let's describe what we observe about the trend of saprobic reproductive morphology. showing the reproductive morphology for several exemplars. Reproductive morphology is usually large, as may be seen particularly clearly in Spathularia flavida, from a sister group of Rhytisma in the Rhytismatales, forming fruit bodies on duff on forest floor. Usually, their outermost layer is mostly naked, with the spore-bearing surface fully exposed, as may be seen particularly clearly in Hederia abetus and Chlorencilia versiformis, which are closely related to Fibrellatsugae, but form saprobic ascocarps on fallen leaves or dead trunks. And usually, the saprobic reproductive body is bright-colored and fresh, almost moist-looking, as may be seen particularly clearly in terrestrial Neolecta species, which together with Tephrina form basal lineages in the Ascomycota. To demonstrate the trend unequivocally, these traits must be placed on a phylogenetic tree, because individual observations of ecology and morphology could be correlated solely or largely due to common ancestry. In other words, perhaps leaf endophyte ecology and reduced reproductive morphology are commonly found together in species just because the species are descended from a common ancestor who had those traits. To ensure that common ancestry was not confounding our conclusion, we characterized the ecology of 77 fungal species as not leaf endophytic, as leaf epiphytic, yellow-green, or as leaf endophytic, green. We included Leiotiomycetes species and close relatives, and also characterized their reproductive morphology as either fully developed, blue, or small and reduced, green. We used the program Bayes traits to integrate across all probable phylogenetic trees based on our rDNA data for these species according to their relative likelihood, assessing whether leaf-living ecology and reduced morphology were evolving independently across the tree or in a correlated manner. The log Bayes factor was nearly 30, revealing very strong support for the correlation, or dependent model, between the leaf-living ecology and reduced fruiting body morphology, validating the trend we have observed as real rather than an incidental consequence of common ancestry. Understanding those relationships between morphology and ecology will be key to continued efforts to describe the genetic basis of both sexual and asexual development. In general, fungal asexual reproductive bodies are very simple and significantly morphologically distinct from the more complex sexually produced fruiting bodies. But for some Leiotiomycetes, such as Halwaya, Ascocorin, and Coxomyces, the asexual reproductive bodies are prominent and composed of tissues homologous to the sexual fruiting bodies. Intriguingly, sexual fruiting bodies of the Rhytismales resemble the simple Conidiomata with which they're physically associated. Some of the same genes or pathways may function pleiotropically in the development of asexual and sexual reproductive bodies in some species. One of the central questions of evolutionary developmental biology is how the same genes and the same pathways often may be activated to perform different developmental functions in different tissues and at different times. Simple morphologies without closely related significant evolved differences limit the usefulness of model fungi such as yeasts and neurospora in exploring the genetic and ecological basis of the development and evolution of intricate morphological structures. Will there be another golden age for the study of evolution and development of fungi after yeasts and neurospora? With many genome projects being completed and new, interesting discoveries being revealed about the constraints and evolvability of morphology and fungi, it seems possible. We hope so.